Hello everyone, welcome to the INDEPTH Academy. Today I'm delighted that Emilia Cepowska is joining us to talk a little bit about her research, and this research that she did as part of an STSM as funded through the INDEPTH cost action. So thanks so much, Emilia, for joining us today. I'm going to let you talk a little bit about your research, then I'm going to come back in and ask you a couple of questions about the STSM. So I'll see you in a little bit.
Thank you. I will talk about my STSM that I did in Tübingen, the place you see on the picture, at Dr. Chang Liu laboratory. And I went there to study the impact of chromatin organization on transcriptional memory in plants. So yes, plants can remember. Okay, plants can remember. It's their evolutionary adaptation to the changing environment, as they are sedentary organisms. So they can adapt to the multiple stress episodes. In our study, we use drought as the stressing factor, and they get more resistant to it. So that means they have to preserve that information about the first stress episode in some kind of form, some kind of memory. But how can plants remember if they don't have brains, right? We observe it as a transcriptional memory in organisms that don't have brains. And it's been already pretty well studied in yeast, but also in plants, focusing mainly on the epigenetic modifications, like histone methylation is responsible for preserving that stress information, or chromatin remodeling. And I focused on, we hypothesize, it's our new hypothesis, that that gene loops can be the marks responsible for stress memory. And already the TF2B factor was shown in yeast to be responsible for formation of gene loops. Gene loops are the chromatin contacts between promoter and terminator. And also it's been shown in yeast to affect transcriptional memory. So we wanted to check if this is the case in Arabidopsis. And I went to Chang Liu to do research on chromatin organization in plants, because he actually shown the organization in Arabidopsis thaliana. For example, he showed that thaliana lacks TADs, the topological associating domains, and also he showed formations like chromatin loops, which we target as our gene loops in stress memory. So I went there to actually learn the 3C methods, chromatin conformation capture methods. 3C is the basic one that allows us to detect the contacts in at the specific loci, and the high C is the most advanced technique that uses deep sequencing to see the chromatin organization genome-wide, and we also will detect our gene loops, and we will compare the results between the 3C and high C. So I performed these two, I learned them, and I performed them there in Tübingen. I adapted the experimental setup from Avramova, where she used drought and to study transcriptional memory. And these are my 3C results. And so the first thing that you would see here, I used memory and non-memory genes to study the effect, but we don't see much differences between memory or non-memory genes. But what we see is that actually in our mutant, the chromatin contacts are increased, which is completely opposite to our hypothesis. And what is important, it corresponds to our preliminary high C analysis that my supervisor is performing. So that's pretty interesting. We don't know still what is the function of this gene loop, so we are investigating that. But I focused on another gene, on one gene that in our lab we already showed the presence of two chromatin loops. First loop goes the 2L primer, it goes in the second exon, and the third loop it goes until the third exon. And here the same, the chromatin contacts were higher in our mutant. And this is the experiment that I actually repeated in Warsaw. So here STSM was very successful to teach me the method, and I was able to introduce it back at my laboratory. And it worked, and the results were also similar. But then when I ran it on the gel, I saw the full gene loop between the 3' and 5' UTR of the gene, which was vanishing in the mutant. So here it corresponds with our hypothesis. Maybe the qPCR was not calibrated perfectly. Maybe I have to still work on that. And to again confirm our mutant, we ran the 3C for FLC gene that is published, that has a function in vernalization of plants. And here also the gene loop forms in this fragment with the 5D primer in the publication. So we detected the gene loop there, and also the mutant showed reduced frequencies of interactions. And that would correspond with our hypothesis, and that gives pretty promising results to use this mutant, not only in stress memory, but to study gene loops function in general in plants. So thank you. I want to thank to my to the team from Świeżewski Lab, to the project supervisor Lien Brzeźniak. You can contact her directly about the project on her email here. And I'm also right now looking for a PhD, so you can contact me on the email here if you would like to talk about that with me. Thank you.
Excellent. Thanks very much, Emilia. So yeah, I should just highlight that this work as part of a master's project that Emilia was doing in the lab there in Warsaw. So you obviously mentioned the STSM. So thanks very much for showing your research. It's really interesting, an interesting hypothesis about how gene loops play a role in generational stress memory there. So first of all, let me ask how this STSM came about. How did you plan to do that?
Actually, it's really connected with the INDEPTH conference that happened in Madrid, where I went to tell about my results with the stress memory, with like the preliminary results with our mutants, only in stress memory. We didn't talk about gene loops, but at this conference, Dr. Chang Liu was there, and I approached him there, and he actually proposed that it's a possibility to apply for STSM, and I did it. I wrote the project and I got it. So actually it's great how many opportunities also conferences and cost action can give us.
That's perfect. It's almost as if I told you exactly to say that. That's exactly what I'd like to see. So yeah, so Emilia applied to come to the conference we had in El Escurial last in December 2019, and obviously met Chang and then went there. So can you talk a little bit more about your STSM? So you went there and you learned to do the high C, is that correct?
Yes. Exactly. I prepared high-C libraries that were later used for sequencing, and the data is not analyzed by me, but by my supervisor. That's really complicated stuff, but I'm also so happy that I could prepare the libraries, and I can be present and observe how to process the data later on the samples that I prepared. So that gives me great grounds to also learn sequencing analysis and more methods. It makes me more curious. So it's really great.
So did you send the seeds to Tübingen first and they grew them for you, and then you went there, or you took the plants with you to Tübingen?
Yeah, I took the plants. I actually prepared this. This was done in our experimental setup that we already optimized in our laboratory. And we used these plants frozen, sent directly to Dr. Chang in Tübingen, and I went there to process them.
Okay, I see. Works beautifully. So I should mention also, as part of the INDEPTH Academy, we actually have a protocol on there, which is for high C, which I worked on with Chang Liu as well, and that's free for anyone to download if you want to learn about how to do high C in Arabidopsis mostly. So that's great. So I think we'll leave it there, Emilia. So thanks very much for your for giving the talk. It's very interesting, and again, you highlight how excellently an STSM can work for training a younger scientist in a new technique, which then you excellently showed that you took back to your lab and it actually worked at your lab. So that's really good.
We have it. Yes, we have it working. It was pretty successful. I'm really happy about that.
Excellent. So this is really great work, and best of luck to you finding a PhD in over there.
Thank you so much. Cool. Thank you.